Good evening, ma'am.
Good evening. So, good evening, everyone. Welcome to today's session of this course plant modeling. So, the last class, we had learned about, we had gone in detail about the FPCB model, the Farquhar von Kammerer Berry model, and what are the three equations and what are the parameters in the equation. Now, in today's class, we will be looking at some ways to, not some ways, a particular way to measure the photosynthetic rate and some details of that and what is the theory behind that. And then hopefully, if time permits, we will start using the plant growth model BioCro, for which I had asked all of you to install R and R Studio so that when I'm doing it, you guys can also uh, have a hands-on session at the same time when I'm also showing, and I'll be going very slowly with the model. So if after the measurement of photosynthetic rate we can move on to the BioPro, we'll be doing that in today's class. So last class, like I just said, we studied the FECB model for solving the ACI curve, and in Farquhar von Kammerer's equation itself, we have this non-rectangular hyperbolic equation which uses some information from the AQ curve. So if we solve the non-rectangular hyperbolic model, we will also get parameters that are corresponding to the AQ curve as well. So these are the main two models that make use of the uh, that are involved in this, even in the software that I was just talking about right now, BioCro. This FPCB model, which also has this non-rectangular hyperbolic model, are the main modules that deal with photosynthetic rate in this BioCro model. So BioCro model is a entire plant growth model that that simulates the entire plant, not just the photosynthetic rate. And for the photosynthetic part of that model, these are the equations that are being used in the BioCoro model. So now today's class we'll look at something briefly about how photosynthesis is measured. Now, in order to solve this equation, I had said that certain parameters need to be measured and they have to be used as input, and certain parameters or certain outputs we can obtain. So, in order to measure these parameters, it is necessary that we measure plants, particularly at leaf scale, to get what is the photosynthetic rate. As of now, photosynthetic rate, we can also get a lot of measurements regarding the productivity of the plant in terms of biomass, the evapotranspiration rate, the stomatal conductance, and many more information from these measurements. But let's see how photosynthetic rate is measured. So in general, if we require to measure photosynthesis, since photosynthesis is a biochemical reaction, the rate of the reaction is what is the information that we require. So it is necessary that we get the rate of this photosynthesis. So rate can be measured in terms of how much carbon dioxide is taken up by the plants. So if you keep a leaf inside a chamber and measure what is the carbon dioxide inside the chamber before the leaf and after the leaf, we can understand that the carbon dioxide which is less because some of the carbon dioxide will be taken up by the plants. So if we measure what is the carbon dioxide that is taken up by the plant, we will get the information about the photosynthetic rate. Another way to get information regarding the photosynthetic rate is by accounting for how much oxygen has been released. So from that information also we can measure the photosynthetic rate. Another way to measure the photosynthesis of a plant is to estimate the dry matter content. Now, when you take a plant, it has leaf, it has roots, it has stem. Dry these parts of the plant, like take a plant and remove its leaf, remove its stems, and dry it, and then weigh it. From this weight, also we can estimate the photosynthetic rate, or we can say that photosynthesis has resulted in this much of dry matter to be produced. So that is also corresponding to a method to uh, quantify the photosynthesis capability or photosynthetic rate of a plant. Same is measuring the carbohydrate production. So that also can be said as an indirect way to measure the photosynthetic rate of a plant. Then another important method is the chlorophyll fluorescence. Again, I won't be going into details of chlorophyll fluorescence, but the chloroplast or the thylakoid membrane has got chlorophyll molecules in the PS2 and PS1 protein complexes, and how effectively they absorb light can be done through this. Uh, absorb light is one basis for this chlorophyll fluorescence, and it can also, this measurement of chlorophyll fluorescence can also be used to estimate or quantify photosynthetic rate. The method that we will be focusing on is the method where leaf gas exchange measurements are obtained. Now, a leaf through its stomata not only takes in carbon dioxide, but it also removes water from the same stomata. So by measuring what is the amount of carbon dioxide that is being exchanged within the atmosphere and the inside the leaf, we can obtain what the photosynthetic rate is. And this method is of obtaining photosynthetic rate is by called measurement through leaf gas exchange, and this can be done through using commercial instruments that rely on the principle of infrared gas analysis. Now, a very commonly used or very familiar instrument is this instrument manufactured by the company called Leica. Leica instruments do and measure leaf gas exchange, uh, taken leaf gas exchange measurements by this principle of infrared gas exchange gas analyzers, and that is how they give us the values of photosynthetic rate, transpiration rate, stomatal conductance, so many parameters related to chlorophyll fluorescence, etc. So in an infrared gas exchange analyzer, what happens is infrared waves are passed through a chamber, and if the chamber has some carbon dioxide molecules in it, this carbon dioxide molecules absorb the infrared rays, and then they measure the infrared rays which is not absorbed. So initially, they know what is the intensity of infrared beams that they are sending into the chamber, and they also know that as a result of absorption, some of the infrared rays are absorbed by the plant. So what is the remaining infrared rays existing in the chamber? It is from this information that it is on the basis of this principle that infrared gas exchange analysis works. So they are going to send infrared waves carbon dioxide absorbs, depending upon the amount of carbon dioxide inside the chamber, they absorb some of the infrared rays. And then when we measure it, what is the infrared rays or infrared infrared remaining in the chamber will help us in understanding how much of carbon dioxide is there inside the chamber. Now, leaf gas exchange or photosynthetic gas exchange systems majorly are of two types. One is the open system, another one is the closed system. So the open system is a system in which um, let me talk about the closed systems first. So closed is in a closed system, what happens is we keep a leaf inside a chamber, transparent chamber, and it is exposed to light. So some amount of photosynthesis happens. Now, initially, when this leaf is kept inside this chamber, it is a closed chamber. Once it is kept inside the chamber, it is not going to get any external input of humidity, or there is nothing to change the carbon dioxide concentration inside the chamber once it is closed. So, whatever was the initial quantity of carbon dioxide, whatever was the initial quantity of water vapor inside it, what uh, inside it, and what happens when the leaf is kept for some time inside it. After some time, water molecules are released by the leaf and some carbon dioxide molecules are absorbed by the leaf. So the net change in the amount of carbon dioxide from the initial and the when measured initially and when measured finally will give us the carbon dioxide uptake by the plant. So this is a very straightforward method. A leaf is kept inside, and slowly the amount of carbon dioxide is going to inside the chamber is going to get reduced because the leaf is going to take up the carbon dioxide. And then when you measure it after a certain point of time, the level of carbon dioxide is less inside this. So what is the difference from the initial and the final value is giving us the carbon dioxide quantity inside the chamber. But one of the I mean, this is not a problem, but one obvious uh, limitation is uh, we are enclosing that leaf away from its natural uh, from its exposure to so many environmental variables that are responsible for photosynthesis. Now, photosynthesis is not something that is only dependent on light. It is also dependent on the water vapor pressure. It is also dependent on carbon dioxide concentration. So all of these variables are cut out when you uh, keep this leaf inside a closed chamber. So in order to have a more realistic uh, measurement values, there is this open system which is continuously exposing the leaf to light, wind, humidity, uh, temperature, and all the environmental variables that is required for this photosynthesis process. And then it is measuring the photosynthetic rate. So this is the main difference between an open system and a closed system. Closed system, you're cutting off all the supply, maybe except for light, because in the absence of light, light reaction won't take place. So light is provided. The chamber is probably transparent, but then no other external inputs are provided, like uh, the leaf is not exposed to a varying relative humidity or wind speed or carbon dioxide concentration or temperature situations. So this limitation is overcome in this open system where a continuous input of air will make sure that the natural conditions of humidity, carbon dioxide concentration is available to the leaf, which is a more realistic representation of the plant while taking measurements. So, open system will look like this. There is a continuous supply of external air from here. This air will go inside this chamber where the leaf is placed, and then the leaf will, when photosynthesis is occurring, leaf will take in some carbon dioxide, leave out some water. That air is again pushed to this chamber, and the air is measured for carbon dioxide and water content. So what was in here will not be here because some carbon dioxide is going to be missing because this plant has taken up the carbon dioxide, and some excess water vapor might be here if transpiration occur. So similar to a closed system, the measurements are similar, but only thing, this continuous supply of air is exposing this leaf to varying conditions of relative humidity and carbon dioxide concentrations, which is replicating the realistic situation much more than in a closed system. So when photosynthetic gas exchange measurements are made, there are two systems, closed system and open system. Closed system is a little limited. The rate measured is limited as a result of no continuous input of relative humidity and carbon dioxide, whereas the open system has a continuous supply of air, which is making sure that it is exposed to the actual conditions of relative humidity and carbon dioxide concentration. Now, in this is how an open system works. Like I said, when air is sent through this, when air is sent to this chamber, initially it is measured for its. I can go here. Initially, it is measured for its flow rate. What is the rate of air flowing inside this chamber? What is the concentration of water inside this chamber? What is the concentration of carbon dioxide inside this chamber? Now, when it goes inside this leaf chamber, leaf is going to take up some carbon dioxide and leave out some water. So, the concentration of water and carbon dioxide, which was inside this, which was given as an input, is going to change because of this photosynthesis and transpiration activity. And then, when this air passes into the sample chamber, we will measure what is the flow rate inside here, what is the concentration of water and concentration of carbon dioxide inside here. And the difference between that will be giving us information about the flow rate for the transpiration rate and the photosynthetic rate. So here, mu E is the flow rate, UE or mu E. UE is the flow rate that is entering the chamber. E represents entry and O represents out. So UO is a flow of air leaving chamber. W is the water concentration entering the chamber. CE is the carbon dioxide concentration entering the chamber. Similarly, WO and CO is the water and carbon dioxide concentration respectively, that is exiting the chamber. This value E is the transpiration rate, A is the photosynthetic rate or assimilation rate, and S is the area of the leaf. Now, the carbon dioxide assimilation can be calculated in this way. When we multiply the flow rate into the carbon dioxide concentration inside the sample minus the carbon dioxide concentration inside this reference chamber divided by the area of the leaf, we will get the rate of carbon dioxide uptake. Similarly, transpiration rate also can be calculated by multiplying the difference in the water concentrations between these two chambers with the flow rate divided by the area of the leaf. So, in order to find out the transpiration rate and the assimilation rate, we will have to do the mass balance of the carbon dioxide, mass balance of air inside the chamber. So right now, since we are interested in the assimilation rate, we are going to do the mass balance of the carbon dioxide concentration inside the chamber. So in order to do the mass balance of the carbon dioxide inside the chamber, we know that the area of the leaf into the assimilation rate is going to give us area into leaf, the assimilation rate will be equal to the flow rate into the concentration of carbon dioxide exiting the system, that is the sample one, minus the flow rate into concentration of carbon dioxide entering the system. So it is the difference in the concentration of carbon dioxide within the output, between the output and the input, that is going to give us the change in storage, like how we do in the mass balance of when we do mass balance. Usually, this is the way we do it. Input minus output is equal to change in storage, mass conservation method. Now, why we are multiplying the area over here is, if you see net assimilation rate, the unit is in moles of carbon dioxide per meter square per second. And the concentration of carbon dioxide is in moles of carbon dioxide per moles of air, moles of carbon dioxide per moles of air. So in order to standardize the unit, we have to, this value, if you multiply it with the, if you multiply it with the area, we will be cancelling out the units to get the concentration value. Now, if you see, I have put this A dash and C naught dash and CE dash over here. And this is because this is representing the dry equivalent of carbon dioxide within the chamber. So when this air enters the chamber, we can measure the water content also. We can measure the carbon dioxide content also. But when we measure the carbon dioxide content, it is important that we consider only the carbon dioxide content. We don't consider the water molecules also which is there inside. So in order to account for that, the carbon dioxide concentration, dry equivalent of the carbon dioxide concentration inside the chamber is represented by Ce by 1 minus We, and outside in the sample chamber, or one that is going out, is represented by Co by 1 minus Wo. So if this is the mass balance equation, when we substitute these three values in A dash, C naught dash and Ce dash, we will get mu naught into C naught by 1 minus W naught minus mu E into Ce by 1 minus We. Again, since SA dash means SA by 1 minus W naught, we will substitute that in place of this in the LHS. And once we solve, we will finally get something like this. SA is equal to W naught into C naught minus 1 minus W naught mu E into Ce by 1 minus We. Now again, when we further simplify it, you can see here
Ma'am, can you please repeat the last step once?
this one where my laser is SA is equal to,
Yes, ma'am. How is
yeah, we are substituting SA dash is equal to SA by 1 minus W naught in the left hand side, this SA dash part. Then we take this 1 minus W naught to the RHS. So it will become mu naught into 1 minus W naught into C naught by 1 minus W naught. This 1 minus W naught is taken into the RHS, so it will come in the numerator. So now we have this 1 minus W naught in the both terms of the numerator. Here, since we have 1 minus W naught in the denominator, this gets cancelled. This term and this term gets cancelled. And then you will get this equation. SA is equal to mu naught C naught minus 1 minus W naught into mu E Ce by 1 minus We. You're just taking this denominator to this to the right hand side. Then moving on, since again this will be further simplified, this 1 minus W naught, which is in corresponding to this term, we will take it is again just represented it more neatly. This will be UECE into 1 minus W naught by 1 minus WE. Now, in order to standardize this, what we do is we multiply and divide by mu E in the RHS. So when you multiply mu E and divide mu E, you will get mu E into mu naught by mu E into C naught minus Ce into 1 minus W naught into 1 minus We. We are actually uh, multiplying and dividing by mu naught, multiply and divide on both sides, in this side by mu naught. So this here, this will be mu naught, this, sorry, mu E, it's not mu naught, it is mu E. So you will when you multiply mu E here and divide mu E, we will get the terms in this way. So finally, we have to arrive at this equation, that is steady state of assimilation rate, and we see that everything is reaching this phase. So what we do is we take down S from the left hand side. We bring the S to the right hand side, so it will come in the denominator part. So we get mu E UE by S into C naught mu naught by mu E minus CE into 1 minus W naught by 1 minus WE. Now this is considered as equal to 1 because the flow rate within the while entering and exiting is assumed as being equal to each other. So in this case, this term will become equal to 1. This will be equal to 1. So finally, we will get this equation. That's the final equation. This equation. So you can this has all these steps for the substitution of the representing values. We just have to take care of these three values. These three values are to be substituted in this mass balance equation. When you substitute this in the mass balance equation and you further simplify it, we will get the answer as assimilation rate is equal to in this following. So this is how assimilation rate is estimated within the instrument. Now, why we have to take water content also? Because again, during the process of photosynthesis, simultaneously this transpiration rate is also happening. Transpiration is also happening. So that is why we, in order to only account for the carbon dioxide concentration without no interference of the water molecules, we have to write the equation in this form. Although if very in simplistic terms, if we don't want to go very complicated, we can use this equation for simple understanding. This is a direct mass balance equation. That is the rate into area is equal to rate into carbon dioxide concentration minus flow rate into carbon dioxide concentration. So that is how we find out the assimilation. That is how assimilation rate is estimated within this leaf gas exchange measuring instrument called LICER, which is working on the principle of infrared gas exchange analysis. Now, this is the instrument that I'm talking about, the infrared gas exchange analyzer. And if you see here, I mean, you can, I've not put in a lot of details. So this is the console, which is giving us all the information, measurement, I mean, whatever measurements can be read. And we can also control the environmental variables using this console part. And here, we can see this is the head of the instrument. In the head, it is somewhere in this part that the leaf is clamped. You can see here this green leaf is clamped within the head of the instrument. So here the leaf will be clamped, and then into this leaf you can provide carbon dioxide concentration, you can provide humidity, you can provide light intensity into this leaf, you can change the temperature, you can change the wind speed. So all of these environmental variables can be controlled. Whatever environmental variables are exposed on a realistic basis, whatever environmental variables a leaf is exposed to can be controlled within this machine itself. So from this machine, you can actually set the temperature so that the leaf is exposed to a particular temperature. You can set the carbon dioxide concentration. You can set the relative humidity. You can set the speed, whatever wind speed it is exposed to. You can also set the light intensity that it is being exposed to. Although you see here that it is closed, we can artificially provide the light that is taken up by the plant. Now, not only can you control the environmental variables, if you're choosing to not control it, the leaf is going to be exposed to air that will be entering the system, and that air will have whatever is the ambient relative humidity and ambient carbon dioxide concentration. All of this can be the leaf can be exposed to the natural conditions as well. Now you see here there is this black thing. If it has a top, so if you remove the top, it will absorb the light from the sun and provide it to the leaf. And similarly, there is a portion where you can where it is there is a tube that helps in taking in air so that this leaf is exposed to the humidity and carbon dioxide concentration which is in the ambient conditions. Now earlier, like how I said, the reference chamber, this is the reference chamber, this is the sample chamber, this is the leaf chamber. So the air is going to enter the reference chamber, and here what are the initial conditions are measured. Then this air is also going into the this air is also being sent to the leaf. This leaf is going to have some photosynthesis, some as a result of photosynthesis, some carbon dioxide, some water addition happens, and this air will then move into the sample chamber. Inside the sample chamber, measurement is done, and when the measurements are done, we'll be able to know what is the amount of carbon dioxide taken in, what is the amount of water that has been added to it. So this instrument and its manual and details about it are all available online. So if you're interested in this part of measurements of the photosynthetic rate, you can go in detail into the manuals, and there are a lot of videos also where they talk about this instrument and how measurements and each and every aspect of what this instrument can be used. I have just given a brief overview of uh, how measurement of photosynthesis is done using this infrared gas exchange analyzer based on the leaf gas exchange, based on the measurement of leaf gas exchange. Now, this is how the instrument make measurements. So, but how do we what do we measure? Now we can't just clamp a leaf and keep looking at the photosynthetic rate. In order to measure what is the photosynthesis rate and what is the respiration rate, we need to follow some protocols, or we need to create an ACI and AQ curve. It is from that we are actually getting uh, now, when you measure it in the ambient conditions, you're only getting the photosynthetic rate for that particular condition, one particular condition. So when the humidity is around 75, it is morning, and temperature is of a particular value, you get the photosynthetic rate for that. But does that represent the photosynthetic rate of the entire plant or entire leaf? No, because the plant is always exposed to different kind of conditions. In the morning, its rate will be different. In the noon, it will be different. In the evening, it is going to be different. Some days when the temperature is low, it is going to be different. Temperature is high, it is going to be different. When the wind speed is high and low, it is going to change. So all these conditions are going to vary. So in order to know what exactly is the or what is the standard photosynthetic rate of a plant of a leaf, we have to measure the ACI and AQ curves for that plant or the leaf or the species. So in order to make this photosynthetic response curve, we can create this photosynthetic response curve using this Leica instrument, and that is done by controlling the environmental factors. Now we know why we are measuring the carbon dioxide response curve. It is the plot that we get with plot of assimilation rate versus the intercellular carbon dioxide concentration. It is giving us the carbon dioxide compensation point, the carboxylation efficiency, the limitations of the stomata can also be obtained from this ACI curves. And then what are the carboxylation limitations? That is in the Calvin cycle, as a result of what is happening in the Calvin cycle, how is the rate of photosynthesis being affected? So all of these can be done, uh, uh, obtained from a response curve. And how do we do the response curve is in the following way. First, we have to set the light to a particular intensity. So normally on an average, say on an average day, if the uh, light that is exposed to plant is say around 1800, then we will set the light to one particular constant value. Say for example 1800. After that, we will change. We keeping all the other factors constant. We'll set the temperature constant. We will also set the humidity to around 50 to 75 percentage. Say we keep it to around 60 percentage, and then we will only change the carbon dioxide concentration. So we set the light to 1800. We set the temperature to a particular value. Say the that day's ambient temperature, say it is 30 degrees Celsius. Then we set the temperature to 30 degrees Celsius. Humidity, generally we set it to 50 and 75 percentage. As a I mean, a lot of experiments have been conducted in a lot of varying conditions. So, usually the humidity is set to a value between 50 and 75. Now, if you don't want to set the relative humidity, say you want to control the vapor pressure deficit of the leaf, you can also do that. But generally, vapor pressure deficit varies with plants. So, for certain plants, the vapor pressure deficit, a minimum or the maximum vapor pressure deficit has to be a particular value, which will change for other plants. So, if you're not aware of that, we can just in general set the relative humidity between 50 and 75. In case of Kerala and all, we set it to around 75 percentage. So after setting humidity, say around 75 or 70 percentage, after setting the temperature to say around 30 degrees, after setting the light to 1800 micromoles per meter square per second, we only change the carbon dioxide concentration. So when we change the carbon dioxide concentration also, there are certain protocols that you need to follow. You see that carbon dioxide concentration, it is first kept at 400 ppm or 400 micromoles per mole. Then it is gradually decreased by 100 micromoles per mole to 300, then to 200, then to 100, then to 50, and then again it is increased to 400, kept at 400 for two or three measurements, and then it is increased to high values of carbon dioxide concentration. Now, why this is done is because at very high, say suppose you were planning to take the CI measurements from a very low value to a very high value, and you keep 50 itself initially, then you keep it at 100, then you keep it at 200. What happens is at very low carbon dioxide value, the Rubisco's activity, the Rubisco can be deactivated. So photosynthesis will stop. It will damage. And then from that time onwards, even if you keep increasing, we might not be able to get the correct rate of photosynthesis. So that is why we keep it at an ambient condition with this all these constant values. We acclimatize this plant to these environmental conditions. Then we gradually decrease the carbon dioxide concentration, and then again we spike the increase to a not a very high value but to the ambient condition, so that again it goes back to the ambient condition, and from there a gradual increase of carbon dioxide concentration is done to a higher value. So if you keep it a very low value or if you keep it a very high value, there are chances that this Rubisco can be deactivated at very low values, and chances are there that the stomata will be close at very high values. Suddenly, if it is exposed to a very high concentration, stomata is going to detect some hazard, and it is going to it is going to close. So then, from then onwards, if you make any measurement, probably you will not get any correct values of photosynthetic rate. So that is why this is one protocol that is followed while measuring the carbon dioxide response curve. You keep it to a light intensity, constant light intensity. That is generally what light intensity does the plant photosynthesis, not the to the maximum intensity that is familiar to the plant. Now, if you set it to 2400 or 3000, probably that is a light limited light limited state. So in those light conditions, the photosynthesis might not increase or decrease. So set the light intensity to around this value, 1800 or 2000, which is usually the maximum intensity the plant works in its best condition, and then set the humidity to around some value between 50 to 75. or if you know in very specific detail about the plant, you can keep the vapor pressure deficit to that value, to a particular value required by the plant, set the temperature, and then change the carbon dioxide in such a way. Similarly, light response curves can also be measured using this IRGA instrument. So again, we all know what are the benefits of making the light response curve. We get the dark respiration rate, we get the light compensation point, we get the quantum efficiency, and also the maximum photosynthetic light saturated photosynthetic rate. Now, this was also helping us, this light response curve. Apart from getting these values, some species of plants are adapted to shaded conditions. It is not very friendly towards very high intensities of light. So, how does this quantum efficiencies and compensation point and all vary for such plants, and how does the compensation, all these parameters vary for plants that are friendly to a lot of sunlight, can also be measured from this AQ curves by comparing these AQ curves. So there are so many advantages of obtaining this light response curve for as per the requirement for various kinds of light impacted studies on photosynthetic rates. Now, similar to carbon dioxide protocols that need to be followed uh, by when we are making carbon dioxide response curve, there are also some light curve strategies. So one strategy that can be used while measuring is the rapid light response curve. So we know that leaves when exposed to light, changes in light, they react very fastly. So first, what they'll do is they will start with a high light level, and then they'll keep reducing it to a lower level. Say they'll start at around 2500, then they'll reduce it to 2300, 2100, 1900, and slowly they will keep on reducing the light intensity. This can be done in the other ways also. You can, adjust the leaf to a low intensity of light, and then slowly increase it to 100, 200, 300, 600, 1000, and keep on increasing from a low level also. And the leaf is exposed to this light condition for some amount of time, say 1 to 2 minutes. The Leica instrument has also this facility uh, where we can input stability conditions. We can say that if stomatal conductance is above this range, then make the measurement. If it is below this range, do not make the measurement, or wait for 3 minutes before taking a measurement. So all these conditions can be set in the instrument itself. So when you expose this leaf to varying levels of light in this order, and leave the leaf for 1 to 2 minutes to get adjusted to that light, and make the measurement of photosynthetic rate, then this method is called the light curve strategy. Now, not a disadvantage, one thing that happens to this rapid exposure, rapid change in light intensity is that although photosynthetic response is quick to light, stomatal conductance or stomatal opening and closing is not very quick to the response of light. So what happens is, even if the photosynthesis amount decreases or increases, the stomata is not going to close or open as quickly as the photosynthetic rate changes. So during that time, if you measure if you see measure the inside carbon dioxide concentration inside the leaf, you will see that it will keep on increasing, because the stomata will not close when it is supposed to close. That is, when photosynthetic rate is decreasing, it is not going to close. So some amount of carbon dioxide will keep entering the leaf, and you will see that the CI value is going on increasing. Now that is not a problem. That is a normal thing that will happen. But again, when we go to check the stomatal conductance data corresponding to this protocol, we might find that it is not a very steady state values or stable values that we get. So in order to overcome that issue where the stomata's response is not reaching a steady state, Only the photosynthetic rate is reaching a steady state, but not the stomata conductance. There is this low light response curve where again you are exposing it to light intervals at say 200 or 300, but you will keep it like that for some 15-20 minutes. So what happens is that leaf and the stomatas in the leaf will get acclimatized to that light condition, and it will reach a steady state. That is, it will reach a state when in that light condition there is not going to be any change in the photosynthetic rate or stomatal conductance for a long time. When the leaf is exposed to that condition, that particular condition, it will reach the steady state of photosynthetic rate as well as the stomatal conductance rate. Then there are other two methods called the survey method and sunfleck and shade method. This is more for trees, which I won't be, more for different particular categories of trees, which we might not need for studies where the degree of shades is going to impact the plants. So I won't be, this is there again in the Likers manuals and information online. There is more details on these methods to be used, and all of this depends on our requirements. So since I'm focusing more on just figuring out the photosynthetic rate for C3 C3 plants at leaf scale, I have used this rapid method to get the light response curve. We can take a 5 minutes break, and next we will be going into the model biochrome. And if anyone has any doubt, you can ask now. Uh, we'll restart the class by 6:52. We'll take a 5 minutes break. Meanwhile, if anybody has doubts, you can ask.
And for this, we have to open RStudio or how it is different.
We'll be running it using RStudio. I'll initially I just want to introduce this model. So right now we don't need to do anything. We can just listen to the class. I'll
Okay.
tell when we can actually do it in the RStudio. Okay. So now we'll go into the application of all of this theory that we studied, these ACI curves, these AQ curves, the FPCB model. How is it that how is all these incorporated, or how is all these information used to model or to estimate photosynthesis using numerical models? Now, we know that photosynthesis is not a very straightforward process. There are a lot of enzymes involved. There are a lot of um, different types of proteins, different types of chemicals, different type of photons are involved, protons are involved, electrons are involved, enzymes are involved. There are a variety of molecules generated. So it's a very complex process. In the in between photosynthesis, transpiration is also happening in the within the stomata with the help of stomata. There is photorespiration also occurring. Now we're talking about C3. Let's leave about the remaining C4 and CAM. So it's a very complex process. So if you go to sit and estimate it manually, it is not going to be easy. So in order to make these estimations, particularly these non-linear equation. Now, like I said, it is not just one process. It is a bunch of biochemical reactions happening. So when all of these reactions are linked with each other, all of these equations are linked with each other, it is not a very straightforward calculation. So we need to make use of numerical methods, processes of iterations, where you over and over and over again you keep on estimating the values, and you will see if it has changed from the previous value. If it has changed massively, then again you estimate with a new set of inputs, and then you will see again if the earlier value and the current value is there is a huge difference, and you will keep on doing this iteration process called iteration over and over and over again, until you reach a point where there is no change in the outputs generated. So that is just a gist of numerical making use of numerical methods to estimate or to solve these equations. The numerical methods are used for solving nonlinear equations. Our system is a very complex system, so we need to make use of complex numerical methods to estimate the photosynthetic rate. That is a combination of a lot of processes together. So that is why we have models. Now, this BioCro model is a plant growth model. It is not only helping us get the photosynthetic rate of one leaf, we get the stomatal conductance of that leaf. We get the transpiration rate of that leaf. We get a lot of parameters from the leaf. We get we not only get it for one leaf, we get it for the entire canopy. And then from the canopy, we know that it is going to grow not just on one day. So from the day of planting, when the germination happens, till the day it is harvested, what is going to be the photosynthetic rate, transpiration rate, stomatal conductance rate, how much of yield it is going to produce. All of this information can be obtained if we put the right equations, if we solve the right equations and find the output of these equations using numerical methods. Now, this model or this software, I would prefer saying it a model, was it was developed in early 1990s, and during that time it was called Vimovac. This was initially developed by S.W. Humphrey and Steve Long. Then over the years, it was updated for this was done for both C3 and C4 photosynthesis. Then it was developed later on. It was rewritten in C program. Then again, for in further years around 2022, they duplicated the code and made it. The basic codes were all in C++, but then they made C to R functions so that we can make use of our program to run this model. The primary developments in recent year includes Ed Scott Road, Deepak Jaiswal, Megan, Steve Long, and Justin McGrath. Dr. Deepak is my supervisor, so he's also one of the developers of this model. Now, like I said, when a plant is growing from the time of its germination till its harvest, there are a lot of processes happening. major process being the photosynthesis process, where carbon dioxide is taken up by the plant. Then uh, there is also this process transpiration, where water is released back into this atmosphere. Water that is taken up by plants from the root, they are released back to the atmosphere. This important process of stomatal conductance, which is the opening and closing of the stomata to let in carbon dioxide and leave out water. Another very important process is carbon partitioning. Now, during photosynthesis, carbohydrates are manufactured. This carbohydrate is not going to be in that one position alone. Whatever carbohydrates are being manufactured has to be sent for the growth of the leaves, for the development of the stem, for the development of the root. So all of this carbohydrate is going to be partitioned into different parts of the plant in different places. So this is called carbon partitioning. This one entire process is also being similar, is also happening in the plant. Then when you see as the plant grows, the bottom leaves are going to be older than the top leaves because new leaves will keep coming. So these leaves will start dying off. That process is called the senescence. So that process is also happening. Then there is root water uptake happening. Water and nutrients are being absorbed by the plant. Then there is the plant is going to get exposed to various kinds of stress factors, like sometimes the soil water in the soil reduces. So as a response to that, the stomata is going to close or open, because there is less water. There is no much water for the plants to conduct this photosynthesis or the leaf energy balance. So that part of the uh, process, or the plant stress as a result of soil moisture reduction can also be simulated using this model. How much water is evaporated from the soil, that is going to impact how much water is going to be available for the plant. So that is also being simulated for the plant. Now, plenty more. I mean, I just picked out a few which we can relate. There are a lot of processes happening in the plant, but majorly we can say this model is able to model the photosynthetic rate, stomatal conductance, transpiration rate, senescence, carbon partitioning, soil evaporation, root water uptake, stomatal water stress because of the soil moisture reduction, and so the development rate uh, uh, by giving some development intakes, that is phenology of the plant, that is at what there is germination, there's flowering, there's reproduction, then there is senescence. All of this is also modeled by this BioCro plant. Now, all of these, when I talk about all of these the photosynthetic rates, stomatal conductance, transpiration, all of these are happening at the level of stomata. So you can imagine, if you take one entire plant, the number of stomata is going to be huge. And these processes are being estimated by this model at the level of the leaf. That is, you measure it for a particular leaf from what is the stomatal conductance, what is the photosynthetic rate, what is the transpiration rate. It is going to be something that is going to be estimated at the leaf. Now, one leaf is going to be different from another leaf. One leaf can be old, one leaf can be new, one leaf can be healthy, one leaf is receiving a lot of light, leaf is in the shade, so it is receiving less light. So based on this segregation, that how much of light is being available to the entire canopy, some portions of the leaves are exposed more to the sun, whereas some portions of the leaves are coming in the shaded part of the plant, so they are not getting light as much as the sunlit portions of the plant is obtained or receiving. So based on that, how is this photosynthetic rate going to change? How is this stomatal conductance going to change? How is transpiration rate going to change? All of this can be done for the entire canopy based on this light interception uh, capability of this BioCro model. So when we first will estimate it for leaf level, and then we will extend that leaf level estimations to the entire canopy based on how much uh, how much light is available to each leaf, or each of to all leaves that are being sunlit, or all leaf which are under the shaded conditions for the plant. So this model is not only doing leaf scale measurement, but it is able to simulate all of this entire, the total photosynthetic rate for this entire canopy. Now, usually, from the background that I am coming, hydrology background and all, the whole, leave alone one plant, a field is, say, a field of rice is represented by probably one rate of photosynthesis, or yield is taken. So one value of yield is taken for that entire field of the plant. Nobody is going to go into details of what is happening at the leaf level, what is happening at the canopy level. So if you see this part, the top part of the canopy, the rate of photosynthesis is different, middle is different, bottom portion it is different. So all of these variations it is a highly dynamic system, and all of these variations to a great extent can be represented by BioCro. Now, BioCro is a modular crop growth model. What do we mean by modular crop growth model? Now, if you we talked about all of these processes. Now, all of these processes are represented by a bunch of equations. We know FVCB equations represent the photosynthetic rate. There is another set of equation called Bolbury equation, which represents the stomatal conductance. Then there is the Penman-Monteith equation, which represents the transpiration. So all these equations, they are written as separate modules. It is not clubbed all together. For photosynthesis, there is one model. For transpiration, there is another model. For stomatal conductance, there is another model. So if we want to study one process alone, we can run that module alone, photosynthesis module alone, to see what is the change in photosynthesis to the environmental conditions. Now, say we want to study stomatal conductance, then we can run the Bolbury module alone to see what is the stomatal conductance happening. Now, suppose we want to see that the stomatal opening is associated with photosynthesis as well as transpiration. So, we want to see what is a coupled response. So, then we will take the stomatal conductance model, we'll take the photosynthesis module, and we'll take the transpiration module. We'll combine it all together, and then we will run it, and then we'll get the combined effect of how all these three processes are impacting each other. This also can be done in BioCro. So generally, usually when coding of a model, or when the process-based model is done, all of it is written as a one whole chunk of code. So it is very difficult for us, say we want to learn individual processes, we cannot utilize that model to segregate the process and study about the plant. But this BioCro model, the modularity nature of this BioCro model is helping us in doing leaf scale measurement for a particular process, as well as doing canopy scale measurements for the entire plant, or with the bunch of processes of our requirement. So that is what we say. That is why we call this crop growth model as a modular crop growth model. So it is similar to when you want to make a customized car, you take you want corresponding this condition or this specification, you assemble all of it, and then you make a car. So it is similar to that. You want to study about say C3 photosynthesis. You don't want to study about how transpiration is happening, you just want to study photosynthetic rate. Then you take that photosynthetic rate, you take the stomatal conductance module, you combine it and run it. You give the inputs necessary, combine it, run it, you will get the output. So that is the modularity nature of this BioCro model. Now, like I said, it is a very physically physical equations, proper equation based process rich model. It is not a statistical model. That is, it is not just playing with data, existing historical data, but it is solving equations to give us the outputs. So that's why we call it a semi-mechanistic model. Then it is a generic model, as in it can be parameterized for any terrestrial crop. Now, I want to study about cassava. So I am going to parameterize it for cassava. So I am going to measure ACI curves for cassava, AQ curves, AT curves for cassava. And then from that measurement, I'm going to get parameters of cassava, and then simulate an entire cassava plant. Similar to that, this model has been developed or has been parameterized for other crops also. One another crop it has been parameterized as soya bean. Similar to that, there is another called bioenergy crops called miscanthus. There is another C4 crop called sugarcane. All of these uh, crops have been parameterized uh, using BioCro. Then, like I said, it is able to do measurements at leaf scale. It can be extended to the canopy scale, and you can also do the measurement for and extend it. This measurement, when you measure it for one canopy, you can extend it to a field scale also. One and the soil water, like I said, soil evaporation is also estimated. How much of soil water is there is estimated using a very simple water balance method. So the advantages again of BioCro in terms of modeling is it's the way it has been coded. There is a C++ basic codes that is then there are C to R objects that is codes to translate the C++ code to R code, and then there is an R interface for us to run the code. Relatively, when you compare it to C++ and Python, R is a little more easier for user friendly, or it is more easier to handle. So running this model using R is I mean is it has become it has the developers have made it in such a way that we can run it in R, which makes it a much more user friendly interface in comparison to C++ and all. Now suppose we don't know R, or we want to develop some modules, say in uh, say we want to represent another process of the plant. So we can itself code it, and then we can put the module in BioPro, and then later on when we are simulating it for the entire plant, you can use that module for that you can combine that module also. So it is not only helping the users to run it, but it is also opening up this opportunity to for us to develop a work on a specific process. Like we say the photosynthetic rate, we want to extend the process. We want to in detail make a module that simulate does the dark respiration rate. Right now the module is not dealing with dark respiration rate, the processes that are involved in it. It is just using one single of dark respiration. Now we want to represent the whole, the citric acid cycle, the glycolysis, all of that process in BioCro. Then we ourselves can write the equations, develop a module, and then put it in BioCro, so that that representation is also correctly done when the plant is being simulated. So in this terms, BioCro stands out as advantageous to a lot of other plant growth models because of the nature in which it has been developed. So I'll again take you to uh, some initial informations regarding BioPro where you can start by reading it. So BioPro software, the online can be, you can just Google BioPro, and the first is my is my screen visible to all of you? Okay. So you can see when you Google BioPro, the first link that comes is this GitHub repository for BioPro. All these codes are open source, so you can download the codes and work with it, and you can it's an open source software. So if you see here, this is talking a little bit of the history. Humphrey and Long started it with the Vimovac, and Lokoi and the remaining developed the BioCro 2, which has been upgraded to BioCro 3. There is one Okay, there is one example to run. Let's not go into that. So BioCro, they are talking about how it can be installed. Simple installation. Now if you want to install it in say Linux, or suppose you're not able to do it the direct installation, they have also given detailed instructions to install it from the source. And in order to make contribution, this is what I said, in order to make develop a module and put it into it, they have the developers manual for it. And then the entire software's documentation is available in this link. So if you go to this link, you go into this page where they have the documentation, all of these, the installation, the overview of the software. Now if you go to articles over here, you can see there is getting started with BioPro. Yeah, the overview and example to run, how to start installation and details about that. So now I would like you take you to a practical guide to BioPro. So this PDF has got good details about starting running a BioPro simulation. When they say running a BioPro simulation, they mean that they are going to simulate it for an entire plant. So full plant growth from the sowing date to the harvesting date, how to run the model. Then in the next chapter has got about how to save the results of BioPro. Then you have module libraries. These module libraries are what I said, are modules that are being the processes that are being represented as modules. So if you go there, uh, okay, I'll show that in the model. So there are several module libraries, and you can access how you can access those module libraries. All can be seen in the uh, this practical guide to BioPro. You can access the source code, how to write a new model. All of those information is there. So right now, let's start by running or just familiarizing with the interface of the I'm not sure if all of you are familiar with R programming, but then again, since time constraints don't allow me to teach you about the basics of R, you are expected to learn that on your own. I'll just tell you how to load libraries, how to run the program, what are the basic the tabs in the R program to run the code or run a line. All of that I'll be telling here. So if you take RStudio, and if you go to a new file, okay, let me just open it, not from here. I'm going to take RStudio from here. And then I'm going to I start a new R script. Now, as soon as we open an R script, what we need to do is see where is this file being worked. I mean, where exactly in this system is this file being worked. So this is the place where we write the code, and this is the console where we are going to, uh, I don't know how to explain it, but yeah, temporary commands can be written over here to just run examples or run temporary lines. You can use this part of the interface. This is the environment interface. So whatever results you get, suppose you run a line, the result will be over here. And this portion you see here, the top, the bottom right corner is where you will get all the help information and everything. So here, the first step I always do is to know where this file is located. Where are we working? So the command for that is get working directory, which is getwd and a set of parentheses, and you just enter it. We see that the file or this new file that I opened is in C users del documents. So this is some random place where it is. I don't want the file to be here. So what I do is I go to um, I go to session tab, and then I go to set working directory, and I'll choose a directory where I want all these files to be saved. So I'm going to choose a location where I want all these files to be saved, and then I will open this. So now we see the working directory, set working directory is in this location. So now when we do get working directory, we see that our working directory has changed from this C users to where I require it to. So that is one step I always will do, because why this is important is, suppose you want to read a file inside this folder, and you're going to run the program in this location. The file is not going to be read. So that is why it is necessary that we always set the working directory to the location where we require it to be run, so that if there are any files or any Excel files or anything that you want to save the outputs to a particular folder, it will go into that folder and not to some random place. So that is the first step I did over here. Now in the getting started part, they are asking us to install this BioPro package. Now RStudio has got a lot of packages in it. So in order to get BioPro, what we'll do is we will install this package BioPro. So for that, you can run this line. If you go here, if you copy this and then paste it over here, you can run this line. Now there is a command called run over here. You can just click on it, and then you will see that this package is being installed over here. So this is the first step you need to do. All those of you are starting, you need to install that package. Now, if you are familiar with Python, there are a lot of packages that will do a lot of things, like matplotlib is there. Then netcdf4 is there that will read netcdf files. Matplotlib is going to help in uh, plotting graphs. So in order to the interface doesn't know, it is just an interface. So we have to say that you have to go to this library and use this library to do these functions. So in order to do that, that library has to be there in the system. So in order to have that library in the system, first we will install this. BioCro is like a library in RStudio. It is a model, but it is stored as a library in the R program. So you can install this first. Now when you see, next when we try to load it, you have to load this library, and the command for that is library. So immediately when you hover the cursor above it, you can see that library, which package you want to load, and then a lot of other arguments, which is not necessary at this point. So you're going to choose library bio, sorry, bi BioCro. See, when we typed bio itself, this BioCro is coming because this has been already installed over here. So modular crop growth simulations, a cross-platform representation of models as a set of equations that facilitates modularity in model building and allows users to harness modern techniques for numerical integration and data visualization, along with where you can find the documentation. They are saying here, if you press F1, you get additional. So you click this and again run it. Now in the keyboard, you can press CTRL plus enter to run it, or you can run the line by using this tab here, run the current line. So now we know that this library is loaded. Now we want to know what all uh, um, okay, I'll just, if you type get here, we know we wanted to talk about what all modules are there in BioPro, what all processes this BioPro can simulate. And for that, it has a function called get all modules. So if you type in get all modules and double quotes, type in biocro. This is a function of biocro alone. You can't type in some other package name here and get all modules of that package. This is a function in the biocro package where we want to get all the modules that are useful in biocro. And then if you run it, see, you see there are around 83 modules that is available in biocro. And let's see over here, ABA decay, that is abscisic acid decay, Ballberry model, that is a stomatal conductance model. Then we have C3 assimilation, what right now we are studying. C3 canopy, that is probably for the entire canopy. Then there is C3 leaf photosynthesis, also something for the leaf alone. You see C4 assimilation there, C4 canopy, C4 leaf photosynthesis. Development index, that is they're assigning an index to every growth stage, phenology of the plant. Then there is FVCB. This is the equation that we studied. FVCB means you can run that FVCB set of equations in BioCro. Then you have their leaf shape factor. We have modules to estimate the light from solar energy, modules to the one layer soil profile that I talked about, how the soil water is estimated. Then there is parameter calculator. So many parameters are there. So some module to calculate all those parameters. Partitioning coefficient logistic. This is what I told. Carbon partitioning. So what are the coefficient? What are the parameters? What parameters have to be used to get the carbon partitioning values? Penman-Monti equations. Those are for evapotranspiration estimation. Then senescence coefficient logistic. That is a module that is going to help in simulating the senescence of the leaves. Soil evaporation, stomata water stress. Then 10-layer C3 canopy. This is again going to separate the entire canopy into 10 layers based on how much of light is absorbed by the plant. So there are a lot of modules that are available separately, which can be used separately to study particular processes in here. Now, I want to, now we got informations regarding all the modules. Now I want to just look into one module level. So one module of interest to us at this point is the FECB module. So what I'm going to do is, there is another function called module info. So now if you type here module, when you type module itself, you get this module info command that comes here. So again, there is a help part that comes up saying that by these biocro module functions, you have to type in this command and put the module name, and then you will get all the information regarding that particular module. So when you're talking about module, I want to go into detail about this FPCB module. So what I do is I copy this and I paste it here. So you have to be careful about these double quotes and all of this. So when get all modules, you need the BioCro package to come in between double quotes. For module info, you need to put in the module name in this format, BioCro colon FPCB. If you want golden ratio hyperbola, whatever is coming here, you can just copy paste it and put it in between this parenthesis, and you will get the information about this module. Now I'm going to run this line, and you see, we get information regarding this entire module. The module name is FPCB. What are the input quantities required? CI is required, intercellular carbon dioxide concentration. G star, that is nothing but carbon dioxide condensation point. J, that is the rate of electron transport. Kc, Michaelis constant for carbon dioxide. K naught, Michaelis constant for oxygen. OI, oxygen concentration, ambient. RD, the dark respiration rate. TPU, the maximum rate of triose phosphate utilization. VC max, the maximum rate of carboxylation. Alpha 2 TPU is a term that is coming while we estimate the triose phosphate utilization limited photosynthesis. Electrons per carboxylation, the values in that equation, the the non-rectangular, when you find out AJ, in the denominator, there is number of electrons for carboxylation, number of electrons for oxygenation. So see, if you give all of these inputs, we will be able to get what is the net photosynthetic rate, AN. What is the carboxylation limited photosynthetic rate? What is the RUBP regeneration limited photosynthetic rate? What is the triose phosphate limited photosynthetic rate? Now let's ignore all these VC, WC, WJ, WP. All of these are parameters that they require for simulating to estimating these four values. So these four values are the values of importance to us. So this module is going to be this module. If you given all of this input, we will get all these outputs. Okay. So I think at this point, I will stop with this. You can run these four codes and go through and do the same thing. Now, this practical guide to BioCro has got so many things here. See, they are saying you have to load this library, you have to load this another library called Lattice. Then another important thing is if you want to know more about commands in here. So you don't know what get module info means. So down here, you can see that I'm typing question mark module info, question mark module info. So what does this module info function or command do? When you do this question mark module info in the right side over here, they will give what does this function do. You can see here, BioCro modules are named set of equation. Each module is available from a BioCro module library. So module info returns essential information about a BioCro module. Similarly, there is another function called quantity list from names. That is a list of named numeric elements from a set of names. That if you want uh, if you want to know where all AN is being used in which all modules this AN value is coming, you can give this quantity list from names. Then evaluate module. You want to run this FVCB module, then you run it using evaluate module command. So suppose you want to run the entire module, then you run it using the run biocro command. So all of this information is available, and what we need to do is to explore it. So first, what you can do as an initial exercise or an initial thing, which I had also asked you to do in the beginning of the session, is to go through this practical guide. Just familiarize with all of this. See, you can see that library is being called here. Install packages is being done to install certain packages. Then there you see here to create a module, print a module, list a module. So many things can be done over here. You can just go through these or you can just, there is one example code that is given here. You can copy this and you can run it and see. That time you will get, so say suppose I just copy this and I run it over here. I'm not going to do anything complicated, just copy the code and I'm going to paste the code here. And then I'm going to run this entire code. I'm going to select this whole thing and run it. After running, you can see I have got a graph over here. Now this graph is giving me what is the stem biomass for the entire with respect to the thermal time, and what is the leaf biomass with respect to the thermal time. Now suppose I want the graph for days of the year. So you can write DOY and you see the plant is growing from the 150th day to something 288 days. That is the Julian days. Julian days is nothing but if it is January 1st, 2001, then that is the first Julian day. January 2nd is the second Julian day. So around 150, it will be sometime around May, and 288 is sometime around August or September. I don't know exactly. That is there. The Julian calendar is available online. So from the 150th day of that particular year to the 288th day, how is the biomass of the stem varying is what you see here in the graph. And how is the biomass of the leaf varying is what you see in the graph. So you can see initially there is very little leaf from the germinated plant. It keeps on increasing and increasing, and towards the end harvesting period, all the leaves are going to diminish and go. Similarly, stem is again increasing very highly, and then towards the end, it will not become zero. There is still some stem remaining. So that is what this graph is telling. So you can familiarize with this code. I will send this code in the WhatsApp group as well as email. I will be uploading it in the eGuru portal as well. So you can just run through it and familiarize yourself with the R program, R Studio. And then in the next class, I will be running this FVCB module. And yeah, that will be our activity in the next. We'll again go through some of the commands in Biochrome regarding the FVCB module, and which we'll deal with in the next class. So I think today that will be it. Thank you for attending the session. If anybody has doubts, you can ask, or you can post it in the Google form that I've shared in the WhatsApp link, the WhatsApp group. Otherwise, you're free to go. Thank you for attending today's session.
Thank you, ma'am.
Thank you.